Oi, boa tarde a todos. Hoje nós teremos um seminário com o Dr. Peter Coleman, que eu não vou apresentar porque ele é um convidado especial, um colaborador, já foi um colaborador do professor João Alexandre, né? E ele que vai apresentá-lo. Então, gostaria de agradecer enormemente já a disponibilidade do Dr. Peter, que nos honra com a sua palestra. E para vocês que estão nos assistindo, vou fazer aqueles lembretes de sempre. Não deixem de curtir a apresentação, de subscrever o canal caso você não tenha subscrito e ativar as notificações, porque isso nos ajuda a subsistir no YouTube. Sempre lembrando que pelo chat é possível fazer perguntas e essas perguntas podem ser feitas em inglês ou em português, porque a gente vai transmitir para o nosso convidado e ao final da apresentação dele ele responderá essas perguntas. Todas as perguntas são muito bem-vindas, todas as perguntas são importantes. Aproveitem bastante o seminário. Então, professor João Alexandre, se pode apresentar o nosso ilustre convidado, por gentileza. Thank you, Peter, for staying today with us.
Boa tarde, pessoal. Eu sou o professor João Alexandre, da Biofísica. Eu vou apresentar para vocês, então, o professor Peter Coleman. Ele é lá da Austrália, de Melbourne. Eu vou falar em inglês para que ele possa entender o que eu estou falando, tá certo? So, switching to English, Professor Peter Coleman is a friend of mine. I was in his lab doing my PhD, so he was a very important person in my scientific formation, and he agreed to give us a talk about his current research, which is in cancer. Now he works at WEHI, Walter and Eliza Hall Institute in Melbourne, Australia. But before that, he has been working for many, many years. As you see, he's a senior scientist, and he is well versed in crystallography, which is his field of expertise, so structural biology. And he has worked since its early beginnings. So he has solved many structures, and he has a big contribution to this area. He was graduated in physics, undergraduate in physics in University of Adelaide, where he also did his PhD. After that, he went to Europe and spent many years there working with very important people. For you just to have a glimpse of what he worked on, there's a paper in Nature from 1976 where he comes after Robert Huber and Johann Deisenhofer. Both of them were Nobel laureates. So that's people with whom he was sharing and learning. So it was about the immunoglobulins. So the very first structures of immunoglobulins were made by him together with these other people. Later on, he worked on viruses, especially influenza virus, and he managed to solve the structure of neuraminidase. He had the help of Joseph Varghese, his colleague at CSIRO in Australia. That's when I met him, was a little bit after this development. And by solving that structure, he actually worked with some chemists as well, and they proposed a drug based on the structure. So what we call drug design or structure-based drug design is actually a successful feature of his curriculum. He put on the market, together with these other people, the drug zanamivir, which is called by GlaxoSmithKline Relenza. So it's one of the drugs available for treating the flu. Okay. He has had many, many awards, and I'll just say that he has got the most important prize in his country for science, and also for civilian citizen. So his contribution to Australia has been in such a size, in such an importance, that he has been awarded both in science and also as a civilian for his contributions, with the most important prizes that there are in Australia. And just to finish on how he has contributed, if you do Google his name in Google Scholar, if you go to Google Scholar, look for citations and things like that, he has over 28,000 citations. His age index is 75 in that particular page of the web. So you can see how he really contributes. On a personal note, he is one of the people that really inspired me. So with that, I will let Peter talk about the BCL2 protein family, which now is his work. It has to do with cancer. And Peter, welcome.
Well, thank you very much, Sean. That's a very generous introduction, very kind of you. It's a pleasure to be with you all here today, and thank you for the invitation to talk about our work. So BCL2 is a funny name for something, but it is nothing more than the second gene to be discovered causing B-cell lymphoma. Back in 1985, it was discovered that patients suffering from follicular lymphoma have a chromosomal translocation that places this new gene, BCL2, under the control of genes that promote antibody production. So BCL2 is an oncogene, but there was a big surprise in 1988 when David Vo, Suzanne Corey, and Jerry Adams, who are now my colleagues at WEHI, and this is at WEHI where they did this work, they discovered that unlike most oncogenes known at the time, which caused uncontrolled proliferation of cells, BCL2 is a gene that keeps cells alive, and it's important in cancer. So the idea of cells dying is a little bit of a strange one, perhaps, until you appreciate that in biological homeostasis, the creation of new cells and the death of old cells is something that is happening all the time. In the blood compartment alone, hundreds of billions of blood cells turn over every day. And unless the same number are created as are lost, then you very quickly have a problem. Insufficient cell death can lead to cancer. Excessive cell death can lead to degenerative diseases. And whilst cancer now has a number of hallmarks, failure of cells to die when they should do so is one of those hallmarks. So I've talked about the BCL2 family. Not long after BCL2 was discovered, a related molecule was found called BAX. And too much of this in a cell, and it dies too easily by apoptosis. Apoptosis had been a form of cell death had been described back in the 70s. And furthermore, in a dying cell, the Bax is found in an oligomeric form. So it is remarkable that BCL2 and Bax are close sequence evolutionary relatives. They share 4 blocks of sequence homology, BCL2 homology regions 1, 2, 3, and 4. And of course, with a measurable degree of amino acid sequence homology, they have very similar 3-dimensional, yet they have completely opposite effects in controlling whether cells live or die. There's a third faction in this BCL2 family, and they're called the BH3 only proteins. It's not a very imaginative name, but it's very accurate because these proteins contain only the third of these 4 sequence homology regions I've talked about. And what the BH3 only proteins are, it appears, is sensors of cellular stress. So these proteins can be upregulated if, for example, the cell's DNA is damaged or if the cell encounters a cytotoxic drug. It's now known that many chemotherapies set off a signaling pathway on the way to killing the drug, which culminates in a BH3 only protein activating the BCL2 pathway to cell death. So three factions. Each faction has a number of family members itself. I'll show you in a minute, but it's interactions between these 3 factions that determine whether cells live or die. And all of this death that we're talking about today happens at the mitochondrial outer membrane. So let me just focus on that piece of the story alone. And if I can just bring up this laser pointer again. Most of these molecules are largely resident on the mitochondrial outer membrane. It's there that Bax and Bac are able to transition from a monomeric form, where they are inert, to an oligomeric form, where they can puncture the mitochondrial outer membrane, releasing cytochrome C and other things into the cytosol. And this sets off a cascade of activation of proteolytic enzymes, caspases, that dismantle the cell. In a healthy cell, Bax and Bac are held in check by an interaction between BCL2 and Bax or Bac. And when a cell senses that it's under stress and needs to die, a BH3 only protein is upregulated and is able to repress this interaction. So the interaction between a BH3 only protein and BCL2 is thought to be able to prevent BCL2 from keeping Bax and Bac in check. The BH3 only proteins release this brake and the cell dies. The BH3 only proteins can also directly activate Bax and Bac. So there are 3-way interactions between the family members. Bax and Bac and possibly Boc, but principally these 2 are the effectors of this cell killing, and they have the 4 BH homology domains. that are identical in their location throughout the structure to those found in the BCL2 faction. BCL2, BCLXL, and MCL1 are among the most important of these so-called guardians. And then there are a number of BH3-only proteins, BIM and BID, and a number of others, some of which are under the control of the P53 protein gene, I should say. So the first structure of this family was determined in the Abbott laboratories in Chicago back in 1996. And it's the structure showing all of the protein but the putative transmembrane domain. We imagine these helical segments to anchor the molecules in the mitochondrial outer membrane, but the part that's in the cytosol is this 8-helix bundle, an N-terminal helix at the back in blue and the C-terminal helices 7 and 8 nearby here. Let me walk you quickly through the fold. The alpha-2 helix shown here contains the important BH3 domain. It's in this part of the protein that the sequence homology block that is shared between all 3 family members is found. Helices 3 and 4 and this central buried helix 5 form a binding groove that I'll show you in a minute, where many of the important interactions take place. Here is that binding groove you can see, and here it is with a BH3 peptide bound to it. The first structure of this type also came from the Abbott laboratories in 1997, not this one in particular, but the first one like this, which gave us the paradigm that the BH3 sequence, which has 4 hydrophobic residues on successive turns of this helix residing in cognate pockets on the surface of BCL2, provide the specificity for the interaction, complemented here by a single salt bridge between an aspartic acid residue and an arginine that's in the BH1 region of the pro-survival protein. So understanding this interaction between the peptide and BCL2 inspired Abbott to discover potent organic ligands that could bind in the groove, just as the peptide does. The first of these they disclosed in 2005, known as ABT737, a modification of that to a clinical candidate called 263. This molecule binds tightly to BCL2 and BCLXL, and the molecule that is currently on the market and in the clinic is ABT199, which is highly selective for BCL2 over the other pro-survival proteins. And this minimizes unwanted side effects from trying to repress the activity of excessive amounts of BCL2 in a cell. So venetoclax ABT199 has been used for treating CLL for several years now, and it's under investigation for other indications, as are other molecules with selectivity for different family members. So cells can be killed by introducing a chemical ligand into BCL2's groove and preventing it from restraining Baxen back. And this just again shows ABT737 in this case, and this is the BIM peptide bound to BCLXL. So that's the first of the interactions. The BH3 binds the groove of the pro-survival protein and kills the cell. How does BCL2 repress BAX? Well, here's a structure of BCL-XL bound to the BAC BH3 peptide, just what would be the alpha-2 piece of BAC. And it binds, it's got a BH3 sequence, hydrophobic residues, a salt link, and all the rest of it. And it all looks the same as if it were a BH3 only peptide. But it requires some unfolding of BAC and BAX if it really is to happen the same way. The hydrophobic surface of this peptide in the groove of BCL-XL is part of the hydrophobic interior of the structure of BAK. So if there's good evidence to think that this is important in the repression of BAC by BCL-XL, but for that to happen, the inert structure of BAC that's being shown here from a crystal structure by others would require some unfolding. And as yet, there is no structure of BCL2 with full-length BAC or BAX, so there are some unknowns around this. Finally, the third interaction, how do the BH3-only proteins activate BAC and BAX directly? Is it also via this helix 3, 4, 5 groove? And this was perhaps the first significant contribution that my colleagues and I were able to make to the field to be able to capture a complex of a BH3 only protein, in this case the BID BH3 bound to BAX. And one reason this had been tricky is that if you just mix the BAX and the BID BH3 or nothing much happens, you can't detect an interaction. But if you mix it in the presence of chaps, the detergent chaps, you notice that this combination dimerizes BAX. So that all seemed pretty interesting. We were able to solve a crystal structure of that, first without the peptide in the groove. The groove is at the back in this picture for reasons you'll see in a minute. But if we put excess BID into the crystallization drop, we could capture the peptide in there as well. So what is this dimer? Well, it's a so-called domain swap dimer. In the monomeric fold of BAX, helix 5 and helix 6 are in a hairpin structure. But in this domain swap dimer, this helix 6, 7, and 8 has come from the partner polypeptide in the dimer. So what does this mean? Well, it does not mean that this dimer is in any way important in apoptosis. At least there's no evidence to suggest it is. But what this structure does tell us is that in the presence of BID and chaps, something we call the latch domain, helices 6, 7, and 8, are able to separate from the rest of the protein that we call the core domain, helices 2, 3, 4, and 5. One seems to be a bit of a floater. So there's the picture showing the front view of the peptide in the groove, and it looks, to all intents and purposes, just like the structure of a BH3 only protein bound to a pro-survival protein, all the same hallmarks, hydrophobic residues, etc., etc. But there's one dramatic difference. This interaction with the pro-survival protein is nanomolar. It's therefore a long-lived complex, and Abbott have shown us, and many others since, that it can be mimicked by drugs. For this interaction, the KD is micromolar, meaning it's transient, and it induces this core latch unfolding of BACs. So one question that we don't yet have an answer to is whether or not this can also be exploited for new medicines, but many people are trying. So this observation about cause and latch separating led us to wonder what might happen to a liberated core domain. So we fused the core domain helices alpha 2 to 5 of BAC to GFP, and we did the same thing for BACs in these papers here, and found that we used the fusion protein GFP because we thought it needed to be chaperoned. We were expecting these core domains to be very hydrophobic. But in any event, even in the presence of the core domain, we found this interesting dimeric structure. Helix 2 of one from the one polypeptide, here's helix 3, helices 4 and 5 are underneath. And here's helix 2 from the other polypeptide, which you can now see is in the 3, 4, 5 groove of the other, just as this is. So this is a symmetric dimer in which you have BH3s of one protein, the alpha 2, in the groove, the 3, 4, 5 groove of the other. These core dimers are highly amphipathic. The surface we look at here of helices 2 and 3 is hydrophilic. The lower surface is extremely hydrophobic. And it made us wonder if this happened in the context of the BACs being anchored to the mitochondrial membrane through its alpha 9, might this not be able to insert into the membrane in the way that a monotopic membrane protein does, so entering into one of the leaflets only of the protein? So I think I've probably been a bit long-winded already, but and I've lost a bit of my screen here, so I'm just going to jump to this panel over here, which just a little movie to illustrate some of the things that we think happens when BAC encounters a BH3. This groove I've talked about in the front is clearly flexible because it's more open when there's a peptide bound, and it's more closed in the absence of a peptide. The peptide binding separates the latch. The alpha 2 becomes exposed somehow. The alpha 1 departs. I talked about it as a floater. There's lots of evidence it's important, but not in the later stages. The BH3 that came in is transiently bound only and leaves, and this freed core domain can form the dimers that I've shown you. And then the surface of this dimer is highly amphipathic. Here is the 4,5 helices with a lot of aromatic residues, which seem ideal for inserting between the head groups of a membrane. So that's the dimer that we think sets off the interaction, but on the dying cells, the BAC and BACs are found as oligomers. And what do those interfaces, how does the protein go on to oligomerize? So there's been a decade or more of research trying to figure this out in the search of a secondary protein-protein interface that would tell us how the oligomer is assembled. And I'll just have to tell you to go and hunt in the literature if you would like to see it. There are indications of some parts of the protein often being close to other parts in the oligomer, but there's no clearly well-defined biochemical interface that leads us to wonder what is the mechanism for membrane permeabilization, since the permeabilization of the membrane seems to be associated with the oligomeric form. Well, just as an aside quickly, there are two different types of membrane pores that are known. The best characterized as the so-called proteinaceous pore, and there's a beautiful recent example of that from a couple of years back in Nature, the Gasdermin A3 structure, where a ring of proteins assemble in the membrane with tight interfaces between the protein subunits. I think in this case, each of these is something like 4 beta strands straddling through the membrane. And then these are connected to each other. There's a very clearly defined protein interface in this assembly in the membrane. And furthermore, the interactions that the protein makes with the membrane are largely, I mean, there are head group interactions, but in this region, it's via the acyl chains of the phospholipids. In contrast, a toroidal lipidic pore is one in which the head groups themselves form part of the lining. It's as if the inner and outer leaflets, or parts of them of the membrane, have somehow fused together, and that the pore itself is lined not so much by the acyl chains of the lipid, but by the head groups, and of course also by the protein. So the question then is, in this situation, should we be thinking about this as an oligomerization of the protein, or would it be better to think of it as a clustering? So for the rest of the talk then, I want to talk about work of one of our students who's, well, he was a student a few years ago, but the work's only just been published, and it was in Nature Structural and Molecular Biology at the end of last year. So if I'm a little bit cryptic in parts of this and you're interested, you could go and look. But the question we're really trying to address is, what more can we learn about mitochondrial outer membrane permeabilization from crystallography or from structural studies generally? And we set about doing this by trying to make the back core domains in the absence of the affinity tag. So to do this, we made it with a GST flag and tag in this time, and we cut that off and we purified the protein. First surprise was that it was soluble, because I was expecting we'd need detergent to keep it soluble. But the size of the purified product after removal of the GST fusion protein suggested we had 6 of these polypeptides in the structure. And the crystal structure indeed revealed a trimer of core dimers. Here are the 3 core dimers from this thing overlaid. They are essentially identical, and you'll recognize from what I showed you before that it's like the earlier structure. That these 3 core dimers are assembled into this ball-like structure with a pseudo 3-fold axis down the center, and facing towards the center are the 4-5-5-4 surfaces of the core dimer, the hydrophobic surfaces, and facing out are the alpha-2, alpha-3 surfaces, the hydrophilic ones. And as I've mentioned, looks pretty much the same as that. But there's a small twist. I don't know how important it is, but there is a slight difference in the way the molecules are assembled in detail, whether it's in the fusion protein or not, just to say that the entity looks to be a somewhat flexible one. Now, I don't know whether you've got the same problem I've got, but I've got my own picture sitting over part of this. I don't know whether I can do something. I should have done that sooner, maybe a long time ago. I apologise for that. So as we were solving the structure, we found that there was density in the maps that was not part of the protein. And indeed, part of it looked like a phosphate or a sulfate ion or something, but clearly too much for just an ion. And indeed, the whole shape of that thing there looked a little bit like a, now I've lost my, let me see if I can, I'm completely blind as to whether you're having trouble seeing this or not, but in any event, this density that we could see reminiscent of a
Peter.
Yes, Sean.
In my screen, which is a big screen, I can see your picture on one side and the presentations on the other side. So at least for me, it's very clear. And
okay, then I'll stop worrying about it. Thank you, Jean. So I think then you can see that density looks to have the hallmarks of a phospholipid head group and the beginnings of acyl chains. So we did an analysis of the purified protein and of the crystals and found that indeed they do contain bacterial lipids, mostly phosphatidylethanolamine. And when we finished refining the structure, we had convincing evidence for the phosphate, the part of the ethanolamine at least, and the glycerol moiety to which the acyl chains are attached, and the first few carbons of the alcohol chains here. The rest of the lipid is disordered in the center of that threefold symmetric ball that I showed you, and indeed there is enough space in there to accommodate what are the 12 acyl chains of the 6 lipids, each associated with one of the 6 polypeptides. So here it is. You can see that the lipids are scarcely visible from the outside, but you can get a glimpse of them. And just to show how they look in the context of a single core dimer, you can see here one phosphate with the 2 beginnings of the 2 chains, and here the other phosphate with the beginnings of the 2 acyl chains. So 3 of these dimeric structures in the ball and all of the acyl chains buried in the middle. The hexamer is a proteolipidic particle, and small angle X-ray scattering sort of confirms that there is something in there. Now, we took this hexamer preparation and recrystallized it in the presence of many different things. We crystallized it in the presence of short chain diacyl lipids, phosphatidylserine and phosphatidylglycerol, in the presence of a monoacyl lipid, lyso-PC, in the presence of a real lipid, phosphatidylcholine, and the detergent C84. And inside all of these structures, we found something that looks like this, a dimer of core dimers. You can see one here, the other one's more or less at right angles to it, in a sandwich-like structure. And again, the surface facing to the center of the sandwich is the alpha-4, alpha-5 surface of the dimer. And it's in there that lipids and detergents also can be detected. I'm not going to talk about these in too much more detail, but there's a lot of crystallographic data in the study. So in all of these different crystals, lipids bind the same site on the back dimers. So here are the structures of at least the head groups of the crystals in which there's phosphatidylethanolamine. This is the original bacterial one, phosphatidylserine, phosphatidylglycerol, and phosphatidylcholine. In every case, the phosphate is hydrogen bonded to the N-terminus of helix 5. So here's the beginning of helix 5 from one half of the core dimer, and here's the C-terminus of alpha 5 from the other half of the core dimer. And there look to also be contributions from here, maybe this tyrosine and arginine are also helping to shape the binding site for the head group. This, I think, is the critical interaction, just because the N-terminus of an alpha helix is such a well-characterized binding site for an anion. And it seems in this case as if in the case of the back core dimers, it is a highly preferred site for a phospholipid to bind. Just to make the point, if I overlay all of these four, the phosphate groups are tightly constrained about where they sit. These structures are at about 2.5 angstroms, most of them, some are better. But there's a common lipid binding site on all of these structures. And the other important point is that the acyl chains, the two different stereochemically different acyl chains, SN1 and 2, are associated with different dimers. So the phospholipid, the phosphate head group is bound at the N-terminus of the alpha 5 of the blue one. The SN2 is also associated with the blue surface, but the SN1 is associated with the magenta dimer, a partner dimer in the assembly, whether it's with an E. coli lipid and it's a trimeric assembly, or whether it's a dimeric assembly, as in the case of the phosphatidylserine. So just to make that point, if we look again at the bottom of the core dimers, here are the phosphates bound at the N-terminus. There's a helix 5 running up there, and here's another one running down here. One phosphate bound here, another one bound here. These guys here, which appear to have acyl chains bound in the gap between helices 4 and 5, this phosphate is bound to the N-terminus of an alpha 5 on a neighboring dimer. I think this whole surface of the core dimers, the alpha 4-5 surface, can accommodate lipids and detergents in all manner of places in the different crystal forms that I've indicated that we have. It's a very sticky, greasy surface, but there does look to be a preferred site for the phospholipid head group itself. So is this real, or is this an artifact? Does this teach us anything at all about how oligomers might form on the membrane. So one way to look at this is to use liposomes to artificial, not really membranes, but this is a mitochondrial lipid mix of a liposome that contains a nickel chelating lipid so that we can attach back. We can't express back with its alpha 9, so to get the back, or to get the full length back minus its transmembrane segment to attach to the liposome, we put a his tag on it and we can stick it on with this nickel chelating lipid. This particular experiment also has in it this modified lipid that contains a photoactivatable group by which we're able to try to cross-link the lipid to any protein that might be nearby. So the experiment is to activate the back on the liposome with a BID peptide. We can get it to activate. We can punch holes in liposomes by this method. But for this analysis, importantly, we can get oligomers of back to form. And then we can fire off, we can give the UV light and try to cross-link the peptide, the lipid, to see where it might be binding, if at all, on back. And that analysis shows that there is a preferred binding site for that particular lipid around residue 105. Residue 105 is right here it's at the beginning of the alpha 4. And if you remember the slide I showed earlier, it is right where the other acol chain was entering. So I think that this experiment has at least hit one of the binding sites, the binding site for the acol chain that's coming from a lipid bound to a neighboring core dimer. So that looks encouraging. We've also done molecular dynamics studies of all of this with our colleagues at La Trobe. I'm not showing the one where they mix the thing with the lipid and see that it assembles in the way I've shown. I'll need to skip that for time, but in the membrane, either using coarse-grained molecular dynamics, as is shown here, or in an all-atom simulation that you can see here hasn't been run for quite as long. But these give the following information. There's a high residency of the phosphate group at the C-terminus of an at the N-terminus of the alpha 5s of the core dimers. So the phosphate binding site is recapitulated. The presence of acol chains between the helices 4 and 5, this was that so-called second site, is common. So there's a strong recapitulation of things seen in the crystal. And in addition, what one sees in the side-on view is that the membrane is thinned underneath the core dimer. And that's just captured graphically here to show that whereas the membrane is typically about 43 angstroms, underneath the protein here, it's only some 28 angstroms. Membrane thinning might be a very important precursor to the fusion of head groups from the outer leaflet with the inner leaflet if you were to make one of these lipidic pores I was talking about. So it looks as if some of the things that we were led to postulate from the crystallography are real when tested in these other ways. Is there anything significant about this for oligomerization? Well, I've made the point, perhaps not as forcefully as I should, but In none of the structures that we've looked at have we ever seen the two acyl chains of a lipid bound to the same core dimer. One acyl chain is always, one is typically bound to the same core dimer as the phosphate head group, but the other one is never associated with that same core dimer. And it raises an obvious question, one that we'd actually thought about, or my colleague Peter Zabatar thought about before we even got this far with the crystallography. But might it not be that the two acyl chains of a lipid, might that not be the way that the core dimers are clustered on the membrane in the way I'm showing here? One acyl chain goes with the phosphate head group into this core dimer, but the other one is available to attach to a second one. So such a pattern of interactions would explain a lot of the puzzling data. Stephanie Blyken and her colleagues have done many beautiful studies showing how these things form oligomers, in particular pores of varying size. I've mentioned that no distinct protein interfaces have ever been found, and my colleagues at WeHi, Ruth Cluck and Rachel Uren, have shown that detergent can disrupt the back oligomers into dimers, which is sort of consistent with a model of the type I'm showing here. So how do you test this idea? And frankly, I don't propose this as a totally rigorous test, but it is a test. We thought we would make the oligomers and then see if we could digest the lipid with a phospholipase to reduce it to oleic acid and a monoacyl lipid to get rid of what you might think of the bivalency of the lipid for these interactions. And we did this first on liposomes, and this is studied just with a blue native page technique. Here you can see the oligomers after you've treated the thing with BID, a laddering of high order things. Here are some controls, but when you introduce the phospholipase over time, you can see that the size of those oligomers reduces. That's nice, but it's only a liposome. More convincing would be to show such a thing happening on mitochondria. And here the experiments get a little trickier and more complicated, because mitochondria being what they are, you really only way you can study the back is by SDS page and Western blotting. And to do all this, you need to do it in a background of back that contains a number of cysteine mutants that can either trap the core dimers or trap oligomers of dimers. Now, this was discovered 10 years ago by my colleague at WeHi, Grant Dewson, and has been a very important analytical tool for us, but it is compromised by the requirement to use mutants. So with all those caveats, here's what happens when you run these things out on non-reducing SDS page and blot them. You can see before there's BID, you have mostly monomeric back. Mostly is perhaps a bit of an exaggeration. There does look to be a strong dimer band in this run, but not much higher order stuff, as you see after you've treated it with BID. And then if we treat the mitochondria now with the phospholipase, we again see that we can reduce that down to the size of a core dimer. And in fact, we know it's a core dimer because of these cysteines that have been used to hold it together. And the lipase has the same effect as the triton. So, so the other way to test whether this is, I need to finish up, I know, would be to see if mutations at the lipid binding site compromised binding. So we've made mutants at the tryptophan and at that tyrosine and arginine and done the experiment on liposomes. Perhaps I'll skip that and jump to the experiment on mitochondria, which is the more relevant one, and point out that when you make the mutation tryptophan 125 to proline, it's a funny mutation, but it's really the only way to get rid of the exposed NH group at the end of helix alpha 5, is to turn that terminal helical residue into a proline. In that case, you can prevent cell killing caused by these two different BH3 mimetic organic ligands. And just to show again that W125P protein, it's capable of advancing to the size of a dimer, is the oligomeric form of the wild type, but it doesn't progress to a form where it's able to permeabilize cells. So that's the end of the story. I think this is, I don't know that anyone's contemplated a mechanism like this yet for oligomerizing proteins on membranes. They may have, but I've missed it. In summary, BAC transitions from an inert state to a membrane active state via core dimers, symmetric dimers of its core domain. These dimers display specific binding sites for lipid head groups and acyl chains. Mutations at the lipid binding site don't affect the formation of the homodimers, but they do compromise membrane permeabilization and cell death. And the acyl chains of lipids can cross-link back dimers and promote back oligomerization and membrane permeabilization. And many people have contributed, but I note in particular Angus Cowan, whose PhD work this was. Angus, he's gone off to Dundee, and my close faculty colleague at Lehigh, Peter Zabatar, from whom I stole most of this presentation from one he gave at the Institute a little while ago. Thank you.
Thank you very much, Peter, for the nice talk. We have some questions from our audience. Let's see. One of our students was asking you if you can tell a little bit more about the molecular dynamics of BAC-dimer evaluation. Can you tell us a little bit more, or once again, how it is evaluated?
Yes, so this is not work I've done myself. It's the work of my colleague Brian Smith and his student at La Trobe University. So I think the part of, perhaps I should have shown the first part of the molecular dynamics as well, but they started out by simply mixing mitochondrial lipids with a BAC core dimer in a box. Throw them all together, just throw it all together randomly in a box. And it quite quickly, it progresses to that structure that I showed at the start. You see the core dimer sitting on the membrane in the way this type of membrane that's called monotopic, because it's only associated with one leaflet of the membrane. it was exactly how we had imagined it might happen when we first saw the core dimers with the hydrophobic surface underneath. So that was quite compelling that happened. And look, you'd need to read the paper to get the details of how quickly that happened. The analysis then becomes one of interrogating many frames of structures over a microsecond or so of molecular dynamics to see how often is there a phosphate where in this place or some other place. And again, there are some numbers given in the paper. How often is there an acyl chain here? The numbers about the membrane thinning are just average numbers over the whole cycle of molecular dynamics. I think it's pretty clear that if you start to draw the acyl chains up into that bottom surface of the core dimer, you will thin the membrane. It's kind of obvious, but the numbers support what seems to be obvious. So we think that all of that somehow is supporting the idea that the core dimer is definitely disturbing the membrane and moving it along the path to puncturing the membrane, but getting to that event is something that we don't see, but we believe that the oligomerization is aided by the diacyl chains of the lipids.
Okay, I think it's nice. It's very nice to imagine all of these things happening. Do you want to make one question, John?
Yes. Peter, thank you for the presentation.
Yes.
Yes, still going into this discussion of how it affects the membrane, because that's the key point when the actual killing of the cell or apoptosis of the cell happens. So from the pictures, one of the pictures you showed, the membrane actually shrinks a bit in the region where the protein is bound. So
yes.
it has a smaller height, and that's because the hydrophobic part of the acyl groups and the phosphates are now bound in a different way towards the more like horizontal with respect to the membrane that is vertical.
Yes.
And then it has a smaller size. Is that helpful for the next step, which is oligomerization of the dimers?
So I don't know. Well, I think that as I've indicated, I think the oligomerization of the dimers is aided by acyl chains of one lipid being drawn up, one being associated with dimer 1 and the other one being associated with dimer 2. So I think drawing the acyl chains out of the bulk of the membrane is one issue. And I think that I'm not saying you can't kill without this happening, but I think it helps killing to happen. I think it's one of the things that promotes the oligomerization. In thinking about, in thinking about other ways, that there's a literature, and you can find some references in our paper to this literature, that if you even put a chemically inert thing, I've just forgotten what the thing was that they used, onto a membrane, they will cluster. And they cluster because anything that is even chemically inert entities can perturb the membrane, and perturbing the membrane is unfavorable. You know, there's an energy penalty associated with that. And you can imagine, I'm waving my hands too much and not giving enough robust scientific explanation, but you can imagine that if you've got two things on a membrane and each of them is paying a price for being there, that price will be lower if they're together. Thermodynamically, they will come together. That's the concept that I think also one needs to keep in mind when trying to understand how the clustering on the membrane occurs. It will just be thermodynamically favorable if the things all migrate to the same point where the membrane is being disturbed.
Okay, so I have another question. You show some residues in the domain that interacts with lipid. Among of them, there is one, arginine 137, I think, that
Yes.
binds to the phosphate part of the lipid. So can you discuss a little bit more about that? Because my group, this question comes from Dr. Marcelo Brigido, with whom I work, and we work with anti-DNA antibodies. And some arginine are pointed in these antibodies as very important to interact with phosphate groups in DNA. So we was wondering if we have somehow the same kind of, interaction between them.
Yes. So the arginine was the other. So I rushed through the mutants a little bit, but you can look in the paper if you're interested. There were 3 residues that we mutated singly and in pairs. And the arginine was the other one that had something of an effect. The tryptophan and the arginine together had the biggest effect. The tyrosine residue, we mutated to phenylalanine or something, I've forgotten. What was it now? Anyway, we mutated the tyrosine and it seemed to be more active. So the problem with making these mutants is that we're making them based on the way they're arranged in the core dimer, but they have a previous life in the inert monomeric fold. Mm-hmm. And it's in that life that if you make the mutant and you have the effect of destabilizing that structure, you might actually promote activation, and that's what we think happens in that case. But as for an arginine binding a phosphate, it's perfectly reasonable, and there would be many cases, I'm sure, where it does contribute, and I think it would be, your DNA binding antibodies would be good examples, and you'd probably find some good examples of similar things also in other DNA protein interactions.
Mm-hmm, mm-hmm. So we have another question from one of our students. She tells something like that. Let me translate a little bit. The interaction with BH3 with BAX inducts modifications, conformal modification in BAX. Was it a challenge to resolve the structure of this complex? That's the question. Yes. It was necessary to make any mutation once the interaction is transient, for example?
Yes. So the challenge in finding any complex between a BH3 only protein and BAC and BAX is that it is so weak. And even in the presence of the chaps, of the detergent, the interaction is still quite weak. And as I mentioned in passing, when we first made the dimer and crystallized it, there was no peptide present. Mm-hmm. And yet without the peptide, you couldn't make the dimer. Uh-huh. So there was some there were some curiosities around all of that, but that needed to be resolved, and I can't say it is all resolved now. I mean, if you were to ask me, why do I need chaps? Why do I need a little bit of detergent to make this happen? I couldn't tell you. All I can say is that in reality, we have done this experiment with a backs and a back that lack the transmembrane anchor. They're just in solution. Mm-hmm. When BID or BIM or whatever BH3 only protein is activating back or backs, the back or backs are on the mitochondrial membrane. Now, does the chaps, I mean, I don't know, I'd be waving my hands, but I'm just pointing out the differences between what we've done and what the real situation is when back and backs get activated and form these core dimers. And some of my colleagues thought that, well, you know, you've got these core latch dimers that you say are irrelevant. You don't want it to end up there. And I'm saying, well, it doesn't end up there because it starts off anchored on a membrane. It opens up, it exposes this surface. There's also there's a paper coming out shortly in Molecular Cell by us in the next little while. It should be available shortly. It's a core dimer structure that includes the latch domains. It's got the rest of the protein attached to it, and it's got some interesting stuff in it. But so the question though was it hard to make the complex? And the answer is not once we realized the trick of needing detergent and the trick of needing excessive amounts of peptide in order to saturate the thing so that we could crystallize the complex. There were no, I think the only other mutants that were in that structure, we typically changed the cysteines in backs to serines just to stop us having to worry about having reducing agents around. But otherwise, that was fairly straightforward.
Uh-huh. Do you want to do another question, Joel?
Yes, if I may.
Yes, we have the microphone, so we got the power.
Peter, if I understood correctly, BH3 is just alpha 2, and it's used by the cell to disrupt the BCL2 back interaction. And by doing that, it allows it to go on forward and induce apoptosis. So Abbott has some molecules which bind to this place, the cleft there.
Yeah.
And I wonder what you guys are doing, because I'm certain that you're thinking about it as well. And how is this actually related to cancer? And how is the view of what are you doing for solving the big problem?
Yes, so I didn't tell any of the story today. We began this work. I began thinking about this problem when I went to Weihai in 2001, because there was a big apoptosis program led by Suzanne Corey and Jerry Adams and Andreas Strasser and David Vo, and people who were deeply embedded in apoptosis research following on the big discovery about BCL2 being a pro-survival protein. So the intention was to see, whether to do effectively what Abbott did. Now they started They were well ahead of everybody. And in 2005, they disclosed their molecule ABT737, and it was just around then that we were having our first successes. We ran high throughput screens, so we discovered molecules not like we did for flu by making substrate analogues of the neuraminidase enzyme. There's an easy place to start with an enzyme. But with a protein-protein interaction, we got started by running high throughput screens. We put together a library of 100,000 molecules, and we screened them to see whether any of them were able to disrupt the interaction between a BH3 peptide and a pro-survival protein. And we had 100,000 compounds, and the molecule that we found had the number 100,308. I don't know whether it really was the last one that went in, but it was, it's probably an irrelevancy. So we did a lot of medicinal chemistry. And when I say we, I don't mean me at all. And we did find molecules that were potently active against BCL-XL, not against BCL2. So BCL-XL is very similar to BCL2, but it turns out that in a platelet, BCL-XL is the protein that determines the life of a platelet. So, there's if you give our molecule to animals, you can see that their platelet numbers dropped quite dramatically. Now they do rebound and all those things, but it's just a way of saying that our program was, I mean, how did we get onto BCL-XL? That was luck more than anything, with just the way the chemistry led us. But those molecules have not advanced into the clinic because most hematologists won't treat patients, cancer patients, with a drug that causes their platelet numbers to drop. So, and this is why Abbott moved from their molecule ABT263 to ABT199, because 263 binds both BCL2 and XL, but 199 is selective for BCL2. So we've done that, and you know, we've played around generally with that groove, looking to see if there are other things we could do. And many drug companies are now playing in this area, and many have drug candidates, even in clinical trials, against others against BCL2, some against MCL1. So this is a very crowded area now of drug company research, and you have to take your hat off to Abbott, who really launched the whole thing and made a success of it. So our interests at the moment are really more in thinking about that other problem. You would think you could also kill a cell if you could tip back and backs over the edge, you know, if you could if you could fire them off directly, then, and so our efforts are really directed at understanding the you know, the structural events that transform back and backs from their inert form to their membrane active form, in the hope that that might suggest different pathways of killing the cell.
Yeah. Oh, so hopefully you can find them.
Well, yeah.
Okay, so I think we can stop now. What do you think? Do you have any other questions, João?
No, it's okay.
It's nice. So, Peter, thank you very much for the nice talk. And we noticed that the sun already rises. Yes. In Australia. So for everybody, it's still 7 p.m. at Australia, 7 a.m. 7 a.m.
It's nearly 8 a.m.
Near 8 a.m. So Peter stayed with us without breakfast, I think. So
or a coffee, definitely.
A little coffee. Okay. So thank you very much for the talk. It was a great honor to have you with us.
Thank you very much. I enjoyed it. Thank you.